Have you ever just stopped and thought about how amazing the human body is? I mean, the way it can heal itself, fix a broken bone, or how you grew from, well, from practically nothing into you.
Yeah,
What's actually behind all that?
it's a great question. And it really gets to the heart of what we're exploring today.
Exactly. Today we're doing a deep dive into stem cells. You could call them the body's master building blocks.
Mhm.
We've got some great source material here from a university biology lecture, and we're going to try and unpack what these cells are all about and, their potential.
Right. The mission here is to go beyond just the headlines you might see. We want to really dig into the core science using the insights from your material. What are stem cells, fundamentally, and what can they do?
Yeah, and some of the stuff in here is pretty surprising, like how all the cells in your body actually come from these precursor cells. But stem cells have these unique tricks up their sleeve, self-renewal, and what's the other one? Differentiation.
That's it. And what's fascinating, truly fascinating, is that while every cell in your body basically has the same instruction manual, the same DNA, stem cells are the perfect example of how different parts of that manual can be read.
Ah, so different genes get switched on or off.
Precisely, leading to different proteins and ultimately creating around 200 different types of cells in your body, each doing its own very specific job.
Okay, so let's really unpack this then. The lecture material defines them by two main things, right? These two key abilities,
Yes, two core capacities. First, there's self-renewal.
which means they can just keep dividing.
Essentially, yes, they can multiply, make more copies of themselves without losing that potential to become something else later. And importantly, without getting old in the cellular sense, they don't undergo senescence. They can divide pretty much without limit.
Wow. Okay, and the second thing?
Differentiation. That's their ability to mature, to change into a specialized cell type.
Like a red blood cell, or a neuron in your brain, or a muscle cell.
Exactly. Becoming a cell with a specific function needed somewhere in the body.
So where do these amazing cells actually exist?
Well, that's a key point in the material. They're found in two main places, in very early embryos and also scattered within adult tissues. But, and this is crucial, their abilities aren't quite the same.
How so?
The stem cells you find in early embryos, they have this incredibly broad potential. They can differentiate into basically any cell type that makes up the organism. It's really extensive.
Okay, so they're like a blank slate that can become anything.
Pretty much. Contrast that with adult stem cells. They can
Mhm.
differentiate, but usually only into cells of the same family, the same lineage.
Right, like you mentioned earlier, the example in the source was a hematopoietic stem cell that makes blood cells, right?
Exactly. It can generate all the different kinds of blood and immune cell, but it's not going to suddenly decide to become a liver cell or a skin cell. It stays within its blood lineage.
That makes sense. It's a more limited, specialized role.
It is. And this difference leads us neatly into this idea of stem cell potency. It's basically a measure of how much differentiation potential a cell has. Your sources actually lay out four categories here.
Okay, let's hear them.
First up is totipotency. Think total potential.
Mm.
A single cell, like a fertilized egg right at the beginning, or maybe the cells just after the first couple of divisions, like the four-cell stage. It can develop into a whole organism, including all the extra bits needed, like the placenta,
The whole package.
the whole package. Then one step down is pluripotency. Pluri means many, right? So these cells can give rise to all the cell types that make up the body itself. But, and here's the distinction, they can't make those supporting structures, the placenta, the amniotic sac, that sort of thing.
Ah, okay. And the source mentioned the inner cell mass of a blastocyst. That's pluripotent.
That's the classic example, yes. That little cluster of cells inside an early embryo. After that, you get multipotency. Multi, meaning several. These cells can develop into a few different cell types, but it's a more restricted range, usually within a specific tissue.
Like the blood stem cells we talked about.
Exactly like those. And finally, there's unipotency. Uni means one. These cells can only differentiate into a single specific cell type. They mostly just self-renew to make more of themselves.
Got it. Totipotent, pluripotent, multipotent, unipotent. That really clarifies the different levels. So how does this link to the main types of stem cells researchers actually work with? The material mentions three key types.
Right, this is where it gets practical. The main players are embryonic stem cells, ESCs, somatic or adult stem cells, ASCs, and a really interesting category called induced pluripotent stem cells, iPSCs.
Okay, so ESCs first. Those are the pluripotent ones.
Correct. They're derived from that inner cell mass of the blastocyst we just discussed. They have that broad potential to become almost anything in the body.
Then the adult stem cells, ASCs, they're the multipotent ones found in our tissues.
Exactly. They're undifferentiated, kind of waiting in the wings within specific organs and tissues. Their main job, day-to-day, is maintenance and repair, replacing cells that wear out or get damaged in that particular tissue.
And the lecture gave quite a few examples of these, didn't it?
It did. It highlights some really important ones, like the hematopoietic stem cells in bone marrow, we mentioned them. They give rise to literally all your blood cell types. Red cells, white cells like B and T lymphocytes, natural killer cells, neutrophils, monocytes, platelets, the whole lot.
That's quite a job description.
It is. Then you have mesenchymal stem cells, also often found in bone marrow. These are the source for bone cells, osteoblasts, cartilage cells, chondrocytes, fat cells, adipocytes, and other connective tissues, kind of the body's structural engineers.
Okay, what else?
Skin stem cells. Makes sense, right? Our skin is constantly renewing. These are found deep in the epidermis and around hair follicles, making new skin cells, hair follicles, keeping that barrier intact. Then neural stem cells in the brain. These can become neurons, the nerve cells themselves, but also important support cells like astrocytes and oligodendrocytes.
Still finding stem cells in the adult brain? That's amazing.
It is. And finally, the example given was epithelial stem cells lining the digestive tract. Think about how much wear and tear that lining gets. These stem cells are constantly replacing the absorptive cells, mucus-producing goblet cells, and other specialized cells there.
So adult stem cells are crucial for keeping us running.
Absolutely vital for day-to-day health and repair. But then there's the third type, which is kind of a game changer.
The iPSCs, induced pluripotent stem cells.
Yes. This is where science got really clever. These actually start out as regular adult cells, say a skin cell. Then, in the lab, they're genetically reprogrammed. Scientists essentially force them to turn on specific genes that are normally active in embryonic stem cells.
So they rewind their development.
In a way, yes. They revert back to a state that's functionally very similar to an embryonic stem cell. They become pluripotent again, able to differentiate into potentially any cell type.
Without needing an embryo.
Exactly. That was this huge breakthrough. It offers a way to get pluripotent cells that are genetically matched to an individual, potentially.
Okay, that's a lot to take in about what they are. So the next logical question is, how do researchers actually get hold of these cells? Where do they come from? The sources touched on this.
Yes, the material lists several potential sources. It mentions fetal tissue, for instance, obtained following an abortion. Also, embryos created for assisted reproductive technologies like IVF that are no longer needed for pregnancy.
Right.
Then there's umbilical cord blood, which is collected right after a baby is born, the blood left in the umbilical cord and placenta. And of course, bone marrow, which is a well-known source, especially for hematopoietic stem cells.
And didn't it also say fetal blood and tissue could be sources for specific types, like neural and hematopoietic stem cells?
Yes, it mentioned those can be harvested from fetal sources as well.
Yeah,
It's important just to lay out what the source material identifies as the origins used.
definitely. And do some sources have particular advantages? The material seemed to highlight a couple.
It did. He pointed out that stem cells from the placenta, similar to cord blood and bone marrow, can be really valuable for treating chronic blood disorders. Things like sickle cell disease, thalassemia, leukemia.
With life-saving stuff.
Absolutely. And it specifically noted an advantage of umbilical cord blood stem cells for transplants. They seem less likely to be rejected by the recipient's immune system compared to, say, bone marrow transplants from an unrelated donor.
Why is that?
The thinking is that these cord blood cells are immunologically quite naive. They haven't fully developed the surface markers that an adult recipient's immune system might recognize as foreign and attack. So potentially safer match.
That makes a lot of sense. Okay, so we know what they are, where they come from. Now, for the really exciting part, what can we do with them? This whole idea of regenerative medicine.
Exactly. This is where the potential becomes really tangible. The idea is using pluripotent stem cells, whether ESCs or iPSCs, as a renewable source to grow replacement cells in tissues to repair damage or treat disease.
And the list of potential applications in the source material is pretty extensive, isn't it?
It really is. It covers a huge range of conditions where cell loss or damage is the core problem. Think about type 1 diabetes, where the body destroys its own insulin-producing beta cells in the pancreas. Stem cells could potentially replace those.
Right.
Or Parkinson's disease. That involves the loss of specific brain cells that produce dopamine. The hope is to replace those lost dopamine-secreting cells. Alzheimer's disease, too, characterized by the loss of neurons due to protein buildup, Could we replenish those neurons?
Neurological conditions seem like a big focus.
They are, because nerve cells don't readily replace themselves. The material also mentions stroke, where brain tissue dies due to lack of oxygen, and spinal cord injuries, which cause paralysis because the connection between the brain and muscles is severed. Replacing damaged nerve tissue is the goal there.
What about conditions outside the brain?
Oh yes. The list includes things like severe burns, where skin stem cells could regenerate skin, heart disease, potentially replacing heart muscle cells damaged after a heart attack, and conditions like osteoarthritis and rheumatoid arthritis, where cartilage or joint tissues are damaged. Could we regrow or repair them using mesenchymal stem cells, perhaps?
It really covers a vast range of unmet medical needs.
It truly does. The common thread is replacing cells that have been lost or damaged beyond the body's natural ability to repair.
Wow. What an incredible journey we've taken through this material. From the absolute basics of what defines a stem cell, that self-renewal and differentiation through all the different types, their potencies, where they come from.
And landing on these really groundbreaking applications in regenerative medicine, it highlights the potential to fundamentally change how we treat so many conditions.
So the big takeaway, the so what from all this?
I think it's the sheer hope embedded in these tiny cells. They represent this incredible, versatile toolkit provided by nature and increasingly resigned by science that holds immense promise. The potential to treat debilitating diseases, repair tissues, maybe even restore function that was thought to be permanently lost.
Truly replacing lost cells, not just managing symptoms.
Exactly. That's the paradigm shift they offer.
So here's something to think about as we wrap up. Imagine that future. A future where your body's own fundamental building blocks aren't just patching things up, but truly rebuilding tissues, organs, offering completely new ways to achieve health and maybe even extend it. Reflecting on everything we've discussed, what aspect of that potential stands out most powerfully to you? Where do you see the path forward?